Cancer. It's a word we're all familiar with, and it's a disease that touches millions of lives. So today, we're going to pull back the curtain and really demystify it. We're going to start big, looking at its staggering global impact. Then we'll journey deep inside our own cells to understand what's actually going wrong. And from there, we'll uncover the really fascinating and sometimes pretty devious role that certain viruses play in this whole process. By the end of this, you're going to have a really clear, fundamental understanding of the biology of cancer.
So here's the game plan for our deep dive today. First, we'll tackle the global challenge to get a sense of the scale. Then we'll get right into the fundamental nature of a cancer cell. After that, we'll explore what are called the hallmarks of cancer, the things that make these cells so tough, and the triggers that start them down that path. From there, we're going to zero in on one crucial cause, viruses. And to wrap it all up, we'll open the case files on some of the most notorious cancer-causing viruses out there.
All right, let's kick things off with a number that really frames the scale of this challenge. Every single year, there are 18.1 million new cases of cancer diagnosed across the world. I mean, just let that sink in. It is an absolutely staggering figure, and what's more, it's a number that's still on the rise.
And of course, the human cost behind those cases is just immense. Cancer is responsible for 9.6 million deaths every year. That makes it one of the world's leading killers and a primary cause of mortality all across the globe.
Now, this chart shows something really important about the global cancer burden. It is not spread out evenly. You can see that almost half of all new cases, we're talking 48.4% to be exact, and more than half of all cancer deaths happen in Asia. This really highlights that this is a complex disease, with genetics, environment, and lifestyle all playing a part.
Okay, so we've seen the massive global scale. Let's shift gears and zoom all the way in. Let's ask the most fundamental question: what is cancer? What is actually happening at the cellular level that causes this disease?
At its very core, cancer isn't just one single thing. It's actually a huge umbrella term for a large collection of related diseases. And the one common thread that ties them all together is that the cells are just not functioning normally. You can think of your body's healthy cells like actors in a really complex play. Each one has a script, they know their cues, they know their role, and everything runs smoothly. Cancer cells, on the other hand, have basically thrown the script out the window. They've gone rogue, and they just start growing and replicating completely uncontrollably, creating chaos.
And this uncontrolled growth leads to something called a neoplasm, which is basically just an abnormal mass of cells. Now, the really crucial thing to understand here is that this growth is relentless. It just keeps going in the same excessive way, even after whatever triggers started the whole process is long gone. It creates a persistent, growing problem inside the body.
So what's the root cause of this relentless behavior? Well, it's an accumulation of genetic alterations, mutations in the cell's DNA. And these aren't just random changes. Each one gives the cell a distinct survival advantage, allowing these neoplastic cells to outcompete their healthy neighbors, to thrive and just multiply.
These genetic advantages give cancer cells what you can almost think of as superpowers. These are specific, acquired abilities that let them break all the normal rules of cell behavior. And to make sense of this, scientists have come up with a brilliant framework called the hallmarks of cancer.
Right, so back in the year 2002, researchers, Hanahan and Weinberg, first pinpointed 6 core superpowers that a cell has to acquire to become truly malignant. These include things like growing relentlessly, which they called sustaining proliferative signaling, ignoring the stop signals, or evading growth suppressors, literally cheating death by resisting cell death, becoming immortal or enabling replicative immortality, creating their own blood supply, inducing angiogenesis, and finally spreading to other parts of the body, which is activating invasion and metastasis. But, science never stands still. By 2011, another decade of research had given us a much deeper understanding, and that original list got an update. This expansion included new superpowers, which they called emerging hallmarks, and a totally new concept called enabling characteristics.
So this is the modern view. It adds 2 more superpowers to the list: the ability to completely reprogram their metabolism to fuel that rapid growth, and the ability to hide from the immune system, our body's own police force. It also introduces those crucial enabling characteristics. Now, these aren't superpowers themselves. A better way to think of them is like a power-up that helps the cancer cells get all their other abilities much faster.
And here they are, those 2 key enablers. First is genome instability and mutation. This basically means the cell's DNA becomes super unstable and just prone to making mistakes, which massively speeds up the rate at which it can get the other hallmark mutations. And second, there's tumor-promoting inflammation. This is where the cancer cell gets really clever and hijacks the body's own inflammatory response, turning it from something that's supposed to protect us into a force that actually helps fuel the tumor's growth.
So, now that we know the superpowers a cancer cell needs, the next logical question is, how does it get them? So now we're going to look at carcinogens, the different internal and external factors that can trigger the genetic mutations that start this whole dangerous process.
These triggers, or carcinogens, basically fall into three main groups. First up, we have the primary, or direct triggers. These are agents like certain chemicals, viruses, or bacteria that directly damage our DNA. Second, you have secondary factors, which are hereditary, things like inheriting a faulty, mutated gene from a parent. And third, there are what we call favoring factors, things like our lifestyle, our diet, our sex, and our age, which can create an environment where cancer is just more likely to pop up.
This chart right here is such a powerful reminder of how much impact our choices can have on our cancer risk. I mean, just look at that. Tobacco is the single biggest cause by a huge margin. The next biggest is being overweight or obese. And this is absolutely crucial information because it means a really significant chunk of all cancers are, in theory, preventable if we make different lifestyle choices.
Let's look at a few specific examples to make this real. A classic chemical carcinogen is asbestos, which is infamous for causing specific cancers of the lung and its lining called bronchogenic carcinoma and pleural mesothelioma. A common physical one is UV radiation from the sun, which is responsible for a staggering 86% of malignant melanoma skin cancers. And in that hereditary category, the only cancer identified so far that's considered purely hereditary is a rare eye cancer in kids called retinoblastoma. Okay, so let's narrow our focus to one of those primary factors we just talked about. Viruses. We're about to explore the fascinating and honestly pretty sneaky ways that certain viruses can hijack our own cellular machinery to cause cancer.
Right now, there are 7 viruses that we know for sure can cause cancer in humans. They fall into 2 main categories based on their genetic material. You've got DNA viruses. Well-known examples are human papillomavirus, or HPV, and hepatitis B. And then you have RNA viruses like hepatitis C, and a special subgroup of those called retroviruses, like one called HTLV1.
The way these viruses cause cancer is through a process called viral transformation. What this means is the virus actually inserts its own genes into our cells. It hijacks their machinery and messes with the normal controls on cell replication. This forces them to grow uncontrollably, lose their normal attachments, and it can even lead to serious chromosome damage. And a key step in this process, almost always, is the integration of the virus's genetic material right into our own DNA. And this brings us to the 3 main strategies or mechanisms that viruses use to pull off this transformation. You've got oncogenic DNA viruses, acute transforming RNA viruses, and slow transforming RNA viruses. So let's break down exactly how each one of these pulls off its cellular heist.
Okay, let's go through these 3 strategies. First up, the oncogenic DNA viruses. They infect a cell that is non-permissive, which means the virus gets stuck and can't finish making new copies of itself. So instead, it integrates its DNA into the host genome. And only its early genes, the ones that scream divide, 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 get switched on. It's like having a gas pedal that's stuck to the floor. The second type is the acute transforming RNA virus. This one is like a Trojan horse. It comes pre-packaged with its own cancer-causing gene, called a viral oncogene, and it just delivers it straight into the cell, causing really rapid transformation. And third is the slow-transforming RNA virus. This one is way more subtle. It doesn't carry its own oncogene. Instead, it inserts its genetic material, including a really powerful on switch called a promoter, right next to one of our own normal growth genes, called a proto-oncogene, and it flips it on permanently, turning our own gene into a cancer driver.
All right, now that we've got the mechanisms down, let's wrap this up by opening a few case files on the key players. We're going to take a closer look at some of the most important DNA tumor viruses and see exactly how they operate. Our first case file is on the human papillomavirus, or HPV. And as you can clearly see from this pyramid, HPV is a massive cause of cancer around the world. It's responsible for over 500,000 cases of cervical cancer alone every single year. So, how does an HPV infection actually turn into cancer? Well, first the virus gets into the deepest layer of the skin or mucosa through tiny little cuts or abrasions. Second, it just hangs out there as a separate, circular piece of DNA called an episome, quietly making copies of itself as the cells grow. Third, in a normal infection, new viruses are just shed from the surface, and that's that.
But the fourth step, and this is the absolute critical event for cancer, is when that circular viral DNA accidentally breaks and wedges itself right into the host cell's own chromosomes. That is the moment it becomes a cancer-causing event.
Okay, next up is Epstein-Barr virus, or EBV. This one is linked to several cancers, including Burkitt's lymphoma, Hodgkin's lymphoma, and nasopharyngeal cancer. EBV's main trick is to transform our immune B cells, and it does this using a key viral protein called EBNA2. You can think of EBNA2 as a master switch that the virus flips to turn on a whole bunch of our cellular genes that drive uncontrolled cancerous growth.
And our final case file is on hepatitis B virus, or HBV, a leading cause of primary liver cancer. HBV is a true master of cellular hijacking. First, it gets into a liver cell and makes its way to the nucleus. Once there, its DNA is converted into this super-stable circular template called covalently closed circular DNA, or cccDNA for short. Our own host cell is then tricked into using this viral template to make viral RNA. And then, in a final clever twist, the virus uses that RNA as a template to make more viral DNA. This creates a vicious cycle of replication and chronic inflammation that can, over time, lead to cancer.
So, we've seen that the deadliest trick these viruses have is their ability to integrate their genetic material right into our own. And this leaves us with a really fascinating, forward-looking question. As our understanding of this process gets more and more precise, could we one day turn this trick on its head? Could we actually harness this mechanism, this ability to insert genes, to develop new targeted therapies for cancer? The answer to that question could truly revolutionize how we treat this devastating disease.